How does your brain send signals so fast? How do your muscles respond instantly when you move? The answer lies in tiny but powerful structures called ligand-gated ion channels. Before we begin, if you haven't watched part 1 of this series, make sure to check it out first. In part 1, we discussed the fundamentals of receptors, including receptor concepts and important receptor theories that explain how drugs interact with receptors to produce their effects. Understanding those basics will make it much easier to follow today's topic. Once you're ready, let's move on to ligand-gated ion channels. In this video, we'll explore what ligand-gated ion channels are, how they work, their structure and mechanism of action, the major types found in the body, and why they are so important in physiology and pharmacology. We'll also discuss their clinical significance and how many important drugs act on these receptors. So, let's begin by understanding what exactly ligand-gated ion channels are. Ion channel-linked receptors, also called ligand-gated ion channels, are special membrane receptors that open or close an ion channel when a specific chemical messenger, a ligand, binds to them. Think of them as specialized gates, closed until the correct key, the ligand, arrives. Once activated, ions rapidly cross the cell membrane, generating an electrical signal. Why are ion channel-linked receptors so important? These receptors are essential because they mediate rapid synaptic transmission in the nervous system, meaning they help nerve cells communicate extremely quickly. Through this fast communication, ion channel-linked receptors play a major role in several critical body functions. They allow neurons to rapidly transmit electrical signals throughout the nervous system, enabling the brain and body to communicate almost instantly. They are also essential for muscle contraction, helping trigger the signals required for movement, from simple actions like blinking your eyes to more complex movements, such as lifting weights. In addition, these receptors contribute to sensory perception, allowing us to process touch, sound, vision, pain, and other sensations from our environment. Furthermore, they are involved in autonomic nervous system function, meaning even automatic processes such as heartbeat, breathing, digestion, and glandular activity depend on signals involving ligand-gated ion channels. Because of their speed and precision, these receptors are fundamental for normal body function and rapid communication between cells. How fast do they work? One fascinating feature of ion channel-linked receptors is their speed. These receptors convert a chemical signal into an electrical signal within milliseconds. For example, think about touching something hot and pulling your hand away before you even realize what happened. That rapid reflex is possible because these receptors transmit signals almost instantly. That's incredibly fast, and exactly why they're essential for immediate responses in the nervous system. How do they work? The process happens in a few simple steps. Step 1. Binding of a neurotransmitter or ligand. In the first step, a neurotransmitter, or ligand, binds to the receptor. You can think of the ligand as a key searching for the correct lock. This binding event initiates the process and leads to the next step. Step 2. Conformational change. Once the ligand binds, the receptor undergoes a conformational change, meaning the protein changes shape. Step 3. Opening or closing of the ion pore. As a result of the conformational change, the third step takes place. The ion channel, or pore, either opens or closes. Step 4. Selective ion movement. With the channel now open or closed, the fourth step begins. Selective ions move across the membrane. Depending on the receptor type, ions like sodium, potassium, calcium, or chloride may enter or leave the cell. This movement in the final step creates rapid electrical changes, allowing cells, especially neurons, to communicate quickly. Let us now understand the general structure of a ligand-gated ion channel. Ligand-gated ion channels are specialized transmembrane proteins embedded within the cell membrane. Most of these receptors are composed of multiple protein subunits arranged together to form a functional channel. In many cases, such as nicotinic acetylcholine and GABA-A receptors. The receptor has a pentameric structure, meaning it is made up of 5 protein subunits arranged in a circular pattern. At the center of this arrangement lies the central ion pore, a channel-like opening that extends through the membrane. This pore forms the pathway through which ions can potentially move across the cell membrane. Although these receptors may appear complex, their structure can be understood by dividing them into 3 main domains: the extracellular domain, the transmembrane domain, and the intracellular domain. First, the extracellular domain. This portion is located on the outer surface of the cell membrane and is also called the ligand-binding domain. It contains specific binding sites designed to recognize neurotransmitters or ligands. When a chemical messenger such as acetylcholine, GABA, glutamate, or glycine binds to this region, it triggers structural changes in the receptor. The extracellular domain is highly selective, meaning each receptor recognizes only specific ligands, ensuring accurate cellular communication. Next is the transmembrane domain. This domain spans across the lipid bilayer of the cell membrane and anchors the receptor securely in place. Most importantly, the transmembrane domain forms the central ion channel, or pore, through which ions move. In many pentameric ligand-gated ion channels, each protein subunit contains 4 transmembrane helices, named M1, M2, M3, and M4. Among these, the M2 segment is especially important because it lines the inner wall of the ion pore and plays a major role in ion movement and channel opening. Finally, the intracellular domain. This domain is located on the inner side of the cell membrane, facing the cytoplasm. Although it does not directly bind ligands, it plays an important role in receptor regulation, channel modulation, and interactions with intracellular proteins. It also helps regulate processes such as receptor trafficking, desensitization, and cellular signaling. Another important structural feature is the selectivity filter, which helps determine the type of ions associated with the channel. Depending on the receptor, channels may be selective for sodium, potassium, calcium, or chloride ions. Thus, the general structure of a ligand-gated ion channel includes multiple protein subunits, an extracellular ligand-binding domain, transmembrane domains, a central ion pore, and a selectivity filter, all organized to form a highly specialized membrane receptor. Now, let's see the types of ion channel-linked receptors. Ligand-gated ion channels can be classified based on the type of ions they allow to pass through the membrane. This classification is important because the type of ion movement determines whether the effect on the cell will be excitatory or inhibitory. The first type is cation channels. These channels primarily allow positively charged ions, or cations, to move across the membrane. Common ions that pass through these channels include sodium, potassium, and calcium ions. In most cases, cation channels produce an excitatory effect because the movement of positive ions into the cell depolarizes the cell. This makes the neuron more likely to generate an electrical impulse and transmit signals. Examples of receptors that function as cation channels include certain acetylcholine and glutamate receptors, which play major roles in nerve communication and muscle activation. The second type is anion channels. These channels primarily allow the passage of negatively charged ions, especially chloride ions. Unlike cation channels, anion channels generally produce an inhibitory effect. When chloride ions enter the cell, they make the inside of the membrane more negative, a process called hyperpolarisation. This reduces the likelihood that the neuron will fire an electrical signal, helping to calm or suppress neural activity. Important examples include GABA and glycine receptors, which are essential for maintaining balance in the nervous system and preventing excessive stimulation. In simple terms, cation channels are usually excitatory because they help activate neurons, while anion channels are usually inhibitory because they help slow down or suppress nerve activity. This balance between excitation and inhibition is crucial for normal brain and nervous system function. So far, we have seen that ligand-gated ion channels can be excitatory or inhibitory depending on the ions they conduct. But what are the major receptor types involved in this process? Let us now look at the different types of ligand-gated ion channels. Let's begin with one of the most important ligand-gated ion channels in the body, the nicotinic acetylcholine receptor, commonly called the nAChR. This receptor plays a vital role in rapid communication between nerves and muscles and is essential for normal movement and nervous system function. The ligand that activates this receptor is acetylcholine, a neurotransmitter released by nerve cells. When acetylcholine binds to the nicotinic receptor, it triggers the opening of the ion channel. Once activated, the receptor allows sodium ions to flow into the cell while potassium ions move out. However, the influx of sodium exceeds the potassium efflux, resulting in a net positive charge entering the cell. This causes depolarisation, an excitatory electrical change that increases the likelihood of generating an action potential. Nicotinic acetylcholine receptors are found in several important locations throughout the body. One major site is the neuromuscular junction, where motor nerves communicate with skeletal muscles to initiate movement. They are also present in autonomic ganglia, helping transmit signals in the autonomic nervous system, and in parts of the central nervous system, where they contribute to cognition, attention, and signaling between neurons. Functionally, these receptors are essential for skeletal muscle contraction. Without them, signals from motor nerves would fail to reach muscles, preventing movement. They are also important for fast synaptic transmission, allowing rapid communication between neurons and target cells. Clinically, the nicotinic acetylcholine receptor has major medical importance. Certain drugs, such as curare and pancuronium, block these receptors and prevent muscle contraction. Because of this effect, they are often used as muscle relaxants during surgery. On the other hand, nicotine, the chemical found in tobacco products, activates these receptors, which is why it stimulates the nervous system. A well-known disease associated with this receptor is myasthenia gravis, an autoimmune disorder in which the body produces antibodies against nicotinic acetylcholine receptors at the neuromuscular junction. This reduces signal transmission to muscles, leading to muscle weakness and fatigue. Another major ligand-gated ion channel is the GABA-A receptor, one of the most important inhibitory receptors in the central nervous system. This receptor plays a critical role in reducing excessive neuronal activity and maintaining balance in the brain. The ligand that activates this receptor is gamma-aminobutyric acid, commonly known as GABA. GABA is the primary inhibitory neurotransmitter in the brain and spinal cord. When GABA binds to the GABA-A receptor, it causes the ion channel to open almost immediately. Once activated, the receptor allows chloride ions to flow into the cell. This chloride influx makes the inside of the neuron more negative, a process called hyperpolarisation. Because the neuron becomes more negatively charged, it is less likely to generate an action potential. As a result, the overall effect of the GABA-A receptor is inhibitory, helping to calm neural activity and prevent overstimulation. GABA-A receptors are widely distributed throughout the brain and spinal cord, where they help regulate mood, sleep, anxiety, muscle tone, and overall nervous system excitability. Their inhibitory action is essential for maintaining normal brain function and preventing uncontrolled nerve firing. These receptors are also extremely important in medicine because several commonly used drugs work by enhancing GABA-A receptor activity. Benzodiazepines, such as anti-anxiety medications, increase the receptor's inhibitory effect and help reduce anxiety and promote relaxation. Barbiturates also enhance receptor activity and are used for sedation and seizure control. Propofol, a powerful intravenous anesthetic commonly used during surgery, acts in part by enhancing GABA-A receptor function, producing rapid sedation and loss of consciousness. The next important ligand-gated ion channel is the glycine receptor, another major inhibitory receptor in the nervous system. Although it is less widely known than the GABA-A receptor, the glycine receptor plays an essential role in controlling muscle activity and maintaining proper nervous system balance. The ligand that activates this receptor is glycine, an inhibitory neurotransmitter primarily found in the spinal cord and brainstem. When glycine binds to its receptor, it triggers the opening of a chloride ion channel. Once activated, chloride ions move into the neuron, a process known as chloride influx. This makes the inside of the cell more negatively charged, leading to hyperpolarisation. As a result, the neuron becomes less likely to fire an electrical impulse. Because of this effect, the glycine receptor is considered an inhibitory receptor, helping to suppress excessive nerve activity. Glycine receptors are mainly located in the spinal cord and brainstem, areas responsible for reflexes, muscle coordination, and many involuntary body functions. The glycine receptor performs several important functions in the nervous system. It regulates motor signals, helping control movement and reflexes. It prevents uncontrolled muscle contractions by reducing excessive stimulation of motor neurons. The receptor also maintains inhibitory control in the nervous system, helping keep neuronal activity balanced and stable. Finally, it controls muscle activity and coordination, ensuring smooth and properly coordinated movements. Clinically, the glycine receptor is significant because it can be blocked by a toxic substance called strychnine. Strychnine acts as a receptor antagonist, meaning it prevents glycine from binding and blocks the receptor's normal inhibitory function. When glycine receptors are blocked, inhibitory control in the spinal cord is lost. This causes neurons to become excessively active, leading to severe muscle spasms, painful convulsions, and uncontrolled muscle contractions. In severe cases, these spasms may even interfere with breathing, making strychnine poisoning life-threatening. Another major group of ligand-gated ion channels is the glutamate receptors. Glutamate is the primary excitatory neurotransmitter in the central nervous system and plays a crucial role in learning, memory, and fast communication between neurons. These receptors stimulate neuronal activity and are classified into several important types. The first is the NMDA receptor, a unique receptor that allows calcium and sodium ions to enter the cell. What makes the NMDA receptor special is that it is both ligand-dependent and voltage-dependent, meaning it requires glutamate binding and specific electrical conditions to become fully activated. One unique feature of the NMDA receptor is the magnesium block. At resting conditions, a magnesium ion sits inside the receptor channel and blocks ion flow, even when glutamate is present. When the neuron becomes depolarized, the magnesium block is removed. This allows calcium and sodium ions to enter the cell, activating processes involved in learning, memory, and synaptic plasticity. In simple terms, the NMDA receptor works only when glutamate binds, and depolarization removes the magnesium block. The second type is the AMPA receptor, which mediates fast excitatory synaptic transmission. When activated, it primarily allows sodium ions to rapidly enter the cell, depolarizing the cell and enabling rapid signal transmission between neurons. The third type is the Kainate receptor, another excitatory glutamate receptor. The Kainate receptor primarily allows the movement of cation ions, mainly sodium influx, into the cell, and potassium efflux out of the cell. This produces depolarization, making the neuron more likely to generate an action potential and contributing to excitatory neurotransmission. In some Kainate receptor subtypes, there may also be limited calcium permeability, but sodium and potassium movement is the primary function. Clinically, glutamate receptors are highly important because excessive activation can become harmful, a process known as excitotoxicity. Overstimulation of these receptors can lead to excessive calcium influx into neurons, resulting in cellular injury and death. This mechanism contributes to neurological damage seen in conditions such as stroke, traumatic brain injury, and neurodegenerative diseases, making glutamate receptors both essential for brain function and important targets in medical research. The next important ligand-gated ion channel is the 5-HT3 receptor, a unique serotonin receptor that differs from most other serotonin receptors because it functions as an ion channel rather than a G protein coupled receptor. The ligand that activates this receptor is serotonin, also known as 5-hydroxytryptamine, 5-HT, a neurotransmitter involved in mood regulation, digestion, and nervous system signaling. When serotonin binds to the 5-HT3 receptor, the ion channel opens, allowing sodium and potassium ions to move across the membrane. This ion movement causes depolarisation, making the receptor excitatory and increasing neuronal activity. The 5-HT3 receptor is found in several important locations, including the central nervous system, where it participates in neurotransmission, and the gastrointestinal tract, where it plays a key role in regulating nausea and vomiting reflexes. Clinically, the 5-HT3 receptor is highly significant because it is a major target for antiemetic drugs, medications used to prevent nausea and vomiting. Drugs such as ondansetron and granisetron work by blocking 5-HT3 receptors, especially in patients undergoing chemotherapy. By preventing serotonin from activating these receptors, these medications effectively reduce chemotherapy-induced nausea and vomiting, greatly improving patient comfort and quality of life. Next, properties of ligand-gated ion channels. Ligand-gated ion channels possess several important properties that make them highly effective for rapid communication in the nervous system. One of their most important characteristics is rapid action. These receptors respond within milliseconds of a neurotransmitter binding, enabling extremely fast signal transmission between cells. Because of this speed, ligand-gated ion channels act much faster than G protein-coupled receptors, which involve more complex intracellular signaling pathways and therefore produce slower responses. Another key property is their short duration of action. The effects of ligand-gated ion channels are usually brief because the channels open only for a short period before closing again. This allows the nervous system to send quick, precise signals without prolonged stimulation, ensuring accurate communication between neurons. These receptors also demonstrate high specificity. Each ligand-gated ion channel is highly selective for the neurotransmitter or ligand that activates it. In other words, only specific chemical messengers can bind to a particular receptor, much like a lock can only be opened by the correct key. This specificity ensures precise control of cellular communication. In addition, ligand-gated ion channels exhibit ion selectivity. Different channels are designed to permit only certain ions to pass through their pores. Some channels selectively allow sodium or calcium ions, leading to excitation, while others mainly allow chloride ions, resulting in inhibition. This selective ion movement determines the physiological effect of receptor activation. Finally, ligand-gated ion channels can undergo desensitization. When a receptor is continuously exposed to a neurotransmitter for a prolonged period, its responsiveness gradually decreases, even if the ligand remains present. This protective mechanism prevents overstimulation and helps maintain balance within the nervous system. In simple terms, the receptor becomes temporarily less sensitive after repeated or continuous activation. Now, let's briefly explore some additional important aspects of ligand-gated ion channels that further highlight their complexity and significance in physiology and medicine. First is subunit diversity. Many ligand-gated ion channels are made up of multiple protein subunits, and the specific combination of these subunits can vary. For example, receptors such as GABA-A and nicotinic acetylcholine receptors may contain different alpha, beta, gamma, or delta subunits. This diversity allows receptors to have specialized functions and tissue-specific properties throughout the body. Another important concept is allostericity modulation. Some drugs and naturally occurring compounds can influence receptor activity by binding to sites different from the main ligand-binding site. For example, benzodiazepines enhance GABA-A receptor activity through an allosteric site, increasing inhibitory effects in the brain. Other substances, including neurosteroids and even alcohol, can also modify ligand-gated ion channel function. Interestingly, ligand-gated ion channels are highly conserved across evolution and are found in both vertebrates and invertebrates. This evolutionary conservation highlights their fundamental role in rapid cellular communication across many species. Although mainly associated with nerves and muscles, some ligand-gated ion channels also function outside the nervous system. For example, purinergic P2X receptors are present in immune cells where they contribute to inflammation and pain signaling. In addition, mutations or dysfunction of ligand-gated ion channels can contribute to disease. These abnormalities are linked to disorders such as congenital myasthenic syndromes, certain forms of epilepsy, and exaggerated startle disorders. Finally, scientists study ligand-gated ion channels using advanced research techniques such as patch clamp electrophysiology to measure ion flow, site-directed mutagenesis to investigate protein structure and function, and pharmacological assays to understand how drugs regulate receptor activity. These methods continue to expand our understanding of how these remarkable receptors function in health and disease. Ligand-gated ion channels offer several important advantages that make them essential for fast communication in the nervous system. One of their greatest advantages is very rapid signaling. Unlike slower receptor systems that rely on multiple intracellular pathways, ionotropic receptors can convert a chemical signal into an electrical response within milliseconds. This incredible speed is especially important for activities that require immediate responses, such as movement, reflexes, and sensory processing. Another major advantage is precise neuronal communication. Because ligand-gated ion channels respond quickly and selectively to specific neurotransmitters, they allow accurate and tightly controlled signaling between neurons. This precision helps the nervous system coordinate complex processes such as thinking, memory, sensation, and motor control. Additionally, ionotropic transmission is highly efficient for reflex activity. Rapid communication through these receptors enables quick protective responses, such as instantly withdrawing your hand from a painful stimulus before conscious awareness fully develops. Despite these benefits, ionotropic transmission also has certain limitations. One limitation is its short duration of action. Since the ion channels open only briefly, their effects are temporary and require continuous neurotransmitter release for sustained activity. Another limitation is limited amplification. Unlike G-protein-coupled receptors, which activate multiple intracellular signaling pathways and amplify responses, ligand-gated ion channels produce more direct but less amplified effects. Finally, excessive activation of ionotropic receptors can become harmful and lead to excitotoxicity. Overstimulation of glutamate receptors, particularly NMDA receptors, can cause excessive calcium entry into neurons, leading to cellular damage or even cell death. This process has been linked to conditions such as stroke, traumatic brain injury, and neurodegenerative diseases. In simple terms, while ionotropic receptors provide fast and precise signaling, their powerful effects must remain carefully regulated to protect the nervous system from damage. Before we end, let us quickly recap what we discussed in this session. We explored ligand-gated ion channels, also called ionotropic receptors, and understood how they mediate rapid synaptic transmission by converting chemical signals into electrical signals. We discussed their structure, mechanism of action, ion movement, excitatory and inhibitory effects, major receptor types, properties, functions, clinical importance, advantages, and limitations. We also looked at important examples, including the nicotinic acetylcholine receptor, GABA-A receptor, glycine receptor, glutamate receptors, and the 5-HT3 receptor, along with their roles in physiology and medicine. In the next part, we will move on to another major class of receptors, G-protein-coupled receptors, or GPCRs, and understand how they differ from ligand-gated ion channels in mechanism, signaling, and physiological effects. Thank you for watching. If you found this explanation helpful, don't forget to like, share, and subscribe for more educational videos on physiology and pharmacology. See you in the next video.